Hi everyone, Miss Calabrese here. Welcome back to our video series about microbiology. So in today's video, we're going to be talking a little bit about the origins of microbes, so how they came to be, and a little bit about how they changed over time. So we're going to talk about two key theories surrounding microbes. All right, so first of all, microbes are ubiquitous, and that just means they are everywhere. So everywhere on planet Earth, you're going to be able to find microbial life. And so that includes, kind of the normal places that you would expect to find it, but also in some really forbidding environments. Microbes have found a way to make a living and survive, even in very, very large numbers. So in places where other things might not be able to survive. So the first life on Earth, we've got evidence of microbial life appearing on Earth. Probably even we can revise this number upward a little bit to about 3.8 billion years ago. So just as the Earth started to cool down enough for life to evolve, it did. So we have microbial life dating way, way back to at least 3.5, maybe 3.8 billion years ago. And for a long, long time, microbes were the only thing on the planet. So those first microbes we could describe as being prokaryotic. So the cells that do not have a nucleus, that don't have any kind of internal organelles, those are the ones that we would have found on the Earth way back then. And it wasn't until about 1.8 billion years ago that we see evidence for eukaryotic life. And eukaryotic life is cells that have a nucleus and have internal cellular structures. And we can tell that by the name there. So the prefix EU means true. The root kary, Kary, means kernel or nucleus. So the true nucleus cells, those happened about 1.8 billion years ago. Right, so here's a real basic timeline of what life on Earth looks like. So again, we can look at where those prokaryotes originate, and we've got evidence of that, right around here, about 3.8 billion years ago, we see prokaryotes. And for a long, long time, it was just them, right? So only prokaryotes on the planet. And it wasn't until about this 1.8 billion year mark that we start to see eukaryotic life. And it's from that eukaryotic life that we start to see multicellular life begin. So we start to see things that have more than just one cell. Up until this point, everything only had one cell. All right, and then eventually we get a whole bunch of different types of critters. So we start to see insects and then things with internal skeletons. We get reptiles, birds, mammals, and then eventually we get primates and humans. All right, so how did we jump from that prokaryotic life, so that cell that didn't have much internal structure, to eukaryotic life, where we do have all these internal structures like nuclei and all of these membrane-bound organelles that we find inside the cell? And the most commonly accepted answer to that question is through the process of endosymbiosis. So we're going to break that word down. So bio means living. Sym means together, and then endo means within. So if we break down this word, it basically means living together within. And that's the root of this theory of how we got eukaryotic life, is that prokaryotes, we had a whole variety of prokaryotic life on Earth. So there's some larger cells, some smaller cells, but they all have pretty simple internal structures. And the way the theory goes is that at some point, a large cell ate a smaller cell, but then did not digest it. So instead of digesting that cell, he just kept that smaller cell alive inside of him, and they lived together, one within the other. And this works because these tiny little cells that were living within the larger cell were beneficial. So they had kind of this mutually beneficial relationship where the little cells that were living inside got protection by living inside that bigger cell, and the bigger cell got a benefit because those little cells were good at doing things. In the case of animal cells like our cells, those little cells became really good at harvesting energy for the big cell, right? So they get this mutually beneficial relationship by living together within one another, right? So that's the kind of origin theory behind eukaryotic life. And it turns out that those little critters that are living within are some of the organelles that you probably recognize from a basic biology class. So you remember learning about the mitochondria, which is the powerhouse of the cell. That mitochondria is actually a prokaryotic cell that's been living inside eukaryotic cells for almost 2 billion years. And same thing is true for chloroplasts. So chloroplasts are organelles that plants have that allow them to be able to harvest energy from sunlight. Those are also prokaryotic cells that at one point were ingested by another cell way back in time and have been living together within ever since. Right, so that's endosymbiotic theory. All right, so let's take it back to just talking about how humans interact with microbes for a little bit here before we get into the next theory that we're going to talk about. So microbes and humans interact in a lot of different ways. So a lot of times when we think about microbes or germs, we usually think of them as kind of stuff that makes you sick. We have to clean them up. They're dirty. They make you ill. But that's not our only interaction with microbes. Obviously, there are a lot of good things that microbes do for us. So we use microbes to be able to bake bread. We use it to produce alcohol, to make cheese. There's even microbial medicine that we use to help to heal people. There are microbial aspects to industry and mining. We can even clean up contaminated parts of the environment by using microbes that are able to process those toxins into something less toxic. So there are a lot of positive interactions between microbes and humans, in addition to some of the ones that we might be more afraid of. All right, so on the scientific side of this, um, the the ways that we use microbes are not always just the pure microbial form. So a lot of times we'll take a microbe and we'll alter its genetics. So anytime we alter the genes of something, that's considered genetic engineering. So it's taking the genes of something, a microbe, a plant, an animal, whatever it might be, and modifying it, right? So that's genetic engineering. A subset of that genetic engineering is called recombinant DNA technology. So recombinant DNA technology is when you take a gene from one organism and you put it into another organism. So for example, if you take a gene from a human and put it into a bacterium, that would be recombinant DNA technology. And that's something that we actually surprisingly do pretty frequently, right? So we're constantly shifting genes around. And we can do that to get microbes to make gene products for us. So the simple example of this would be if you think about diabetics who need to take insulin. Historically, insulin that was needed to be injected by diabetics came from the pancreases of pigs. So we would harvest pigs and use their pancreases to make insulin and then give that insulin to people so that they could inject it to control their blood sugar. But that, as you can imagine, is pretty inefficient and it can lead to some problems if that pig insulin doesn't work quite as well as human insulin. So we use recombinant DNA technology to take the gene for human insulin, put that gene for human insulin into a bacterium, E. coli, and get that E. coli to make actual human insulin for us. And then we've got this perfect human insulin that was manufactured by bacteria. So that's an example of recombinant DNA technology. Right. And then another human use of microbes is bioremediation. So that's taking microbes that can process toxins and convert those toxins into something less toxic and more environmentally friendly. Okay, so those are good interactions we have with microbes, and then there are also bad ones. So the microbes that can cause infectious disease, those ones we refer to as pathogens. So pathogens are microbes that cause disease. And obviously, microbial disease is something that's a huge problem all over the world. There's lots of infectious disease that's being spread all the time um, by lots of different types of microbes. We are lucky to be living in a time where we have lots of functional antibiotics and lots of functional vaccines that prevent a lot of death from infectious disease, but there's still quite a bit of that out there. In fact, there are diseases that we still don't have vaccines or treatments for that are very deadly. So, for example, on this slide we're talking about malaria, which is a microbial disease that that causes a lot of unnecessary death just because we don't have the ideal ways to treat people with that disease. All right. A quick note about emerging diseases here. And so I usually talk about emerging diseases in the context of these are diseases that seem to kind of show up out of nowhere. Emerging diseases we consider them emerging because they usually existed previously, but then jumped to a human host. So remember that everything can get infectious disease. So plants get diseases, animals get diseases. Those diseases don't always stay in their host species. Sometimes they can jump to new hosts. And when they jump from animals to humans, they look like an emerging disease to us. So, for example, historically we would talk about things like Ebola or HIV, and those things that jumped from species to humans. And then once they become spread in humans, then they can cause some pretty serious disease. The obvious one that's spreading around the world right now very rapidly is the coronavirus. So that coronavirus existed before, right? Coronaviruses are common viruses in bats. In fact, there are some strains of coronavirus that cause the common cold, but this current one just jumped from a bat to a human not that long ago, and was able to become incredibly infectious and have a very high mortality rate, which is why we're dealing with this problem the world over right now. So why are there so many emerging diseases? Usually, it just comes from humans interacting very closely with animals, and the reason that we interact closely with animals is usually to eat them, unfortunately. So animal agriculture, wet markets like the ones where a whole bunch of different species of animals are aggregated all together, creates a perfect breeding ground for microbes to jump from one species to another and possibly then result in community spread, which is what's happening with the current COVID-19. All right. So even some things that we did not historically think had any microbial origin, turns out there might be some actual microbe at the root of some of these conditions. So gastric ulcers, so stomach ulcers, these are people didn't used to think there was any microbial origin. They thought, okay, you have too much stress, drank too much coffee, drank too much alcohol, then you would get ulcers in your stomach. Turns out those are contributing factors, but the main factor is the presence of a bacterium called Helicobacter pylori. And if you eliminate the Helicobacter pylori, then you can get rid of that stomach ulcer. There's some evidence that type 1 diabetes is caused by exposure to a virus called the Coxsackie virus. So that when infants are exposed to this virus in the first months of life or the first couple of years of life, then they're more likely to develop type 1 diabetes later in their childhood. There's even some evidence that mental illnesses like schizophrenia, like obsessive compulsive disorder, depression, that these can be linked to viruses or bacterial content of the gut. So we're learning a whole lot about infectious diseases that maybe you wouldn't normally think were causing these kind of issues. All right. So real quick about understanding where microbes come from. So there used to be a long, long time ago, a theory that life just came out of nowhere. And so this would, if you can imagine, if you had some food that you left out on the counter and you came back to that food a few days later and you noticed it had mold on it, or it had maggots on it, or it turned gross, right? The theory was that life, that mold, maggots, whatever it was, just came out of nowhere, spontaneously generated on your food. That theory that life comes out of nowhere was called abiogenesis. So genesis meaning origins, bio meaning life, and a meaning not. So life coming from not life. So just coming out of nowhere. That's not true. So we disproved that theory. And so the current theory is that life comes from life. So that theory is biogenesis, that life comes from life, a life originates from life, meaning that those maggots did not appear out of nowhere on your food. They came from microscopic eggs that were laid by a fly, and you just couldn't see them. They were too small to be seen until they had grown into maggots. Same thing with that mold. There were tiny invisible mold spores that landed on that food, and that grew into larger, more visible structures over time. Right, so we disproved the theory of abiogenesis and replaced it with the theory of biogenesis. All right. So let's talk a little bit about how we're able to visualize microbes. So we use microscopes. Essentially, we use microscopes to look at microbes. Microscopes, the first kind of very, very basic magnifying lenses were developed by a scientist called Robert Hooke. So he developed a magnifying glass that allowed him to look at things slightly larger than they actually were. But then the real advancement came with another scientist called Leeuwenhoek, and he developed a microscope. Actually, he was a tailor. He developed a microscope so that he could do thread counts of the fabrics that he was working on. So he wanted to be able to magnify and look at the threads in his fabrics. Then he thought, well, why not look at some other stuff? Why not look at pond water? Why not scrape my teeth and see what's growing on there and look at all of these microscopic things? So somehow this tailor who decided to be creative and come up with a microscope became kind of the father of microscopy because he was the first one to sort of explore this microbial world. All right. Another scientist we should be familiar with is Louis Pasteur. And Louis Pasteur, among a lot of other things, one of the things he did was to actually disprove that theory of abiogenesis. So back in the 1800s, his scientific work led to us creating the theory of biogenesis. And his experiment that he used to come up with this theory, a lot of times just we call it the swan neck flask theory, is visualized in this picture here, where he took two flasks that he put broth in them. He boiled the broth as a way of sterilizing it and killing anything that might be in there. And then he just let the broth sit out. One of the flasks had a swan neck, so a neck that was kind of like this one here. And you can imagine because the flask had a neck like this, it would be difficult for particles in the air to actually land in this flask, go all the way down the neck of the flask and find its way into the broth. Um, the, the other flask he left open to the air, so he didn't have a swan neck on this other flask. And what he found was that even though the broth in these flasks was identical, like he boiled them, they were both as sterile as they could have been at the time. Um, Once he left them out for several days, you can imagine that this one, the one that was open to the air, turned gross faster. The broth became cloudy, it became stagnant, its stuff started growing in there, bacteria, you can imagine that it started to smell. Whereas this other one, the one with the swan neck, um, particles that were in the air, so tiny bacteria, whatever, mold spores that might have been floating in the air, even if they would go into the neck of this flask, they would kind of get stuck in this region here and not make it all the way into the broth. So it took a lot longer for that swan neck flask to show signs of growth than for the non-swan neck flask. And so this was Pasteur's way of saying, listen, the life didn't come out of nowhere. If the life came out of nowhere, then we would have seen growth in both flasks equally, but we didn't. We only saw it in the one that was open to the air. So that's a hint that there is invisible life in the air that we just couldn't see. Right? And so that led to a lot more research on microbes and how they are transmitted through the air. Right. Another scientist we should be familiar with is Robert Koch. So Robert Koch um, came up with our germ theory of disease. So he came up with a series of rules that he used to associate one particular germ, one pathogen, whether it's a virus, a bacterium, whatever it is, to associate with that one microbe with a disease. Right, so and he was the first one to kind of come up with actual rigid rules to be able to do that so that we could say, okay, if you have these symptoms, if we think you have this particular disease, that it must be caused by this one particular germ. Right? So the basics of what we refer to as his postulates are that if we take an animal that's sick and we extract the pathogen that's making that animal sick and we grow it in culture, so grow pure culture of this particular bacterium, and we take that bacterium and inject it into a healthy animal, that healthy animal will come down with the same exact disease, and that we'll be able to again isolate that bacteria from that now ill animal, right? So to prove that one particular pathogen is what caused that line of, or what caused those symptoms, what caused that disease in that particular animal, right? So this is the first time anyone had come up with kind of like a rigid scientific way to logically prove that one germ causes one particular disease. All right, so just a couple questions here to check that we understand the concepts. What is endosymbiotic theory? And then what is germ theory? All right, and here's our answer. So endosymbiosis literally means living together within. So endosymbiotic theory is this theory that eukaryotic life originated from prokaryotic life symbiotically living together. So one prokaryote lives within another and becomes an organelle now within that now eukaryotic organism. And then what is germ theory? That's the theory that infectious disease is caused by microorganisms. We can trace one germ to one disease. All right, so I hope this was helpful. Let me know if you have questions.